I'm Daniel Levine, and this is the Bio Report. Systemic chemotherapy remains the foundation of cancer treatment, but its widespread toxicity often cuts short potential therapeutic benefits. NanOlogy is developing a new approach, localized cancer therapy that keeps the drug where it's needed most. We spoke to Marc Iacobucci, managing director of NanOlogy, about the company's precision particle engineering platform, how it transforms existing oncology drugs into microparticles optimized for intratumoral delivery, and how this enables sustained high-concentration dosing inside tumors that destroys cancer cells, stimulates immune responses, and spares patients the debilitating effects of systemic chemotherapy. Marc, thanks for joining us.
Danny, thank you too. Really appreciate the opportunity to participate in Bio Report podcast with you.
We're going to talk about cancer, NanOlogy, and its platform technology designed to optimize intratumoral drug therapy to improve solid tumor response and minimize serious toxicities. Let's start with systemic cancer therapies, though. What are the problems with systemic delivery?
Well, when you learn that a loved one is diagnosed with cancer, one of the first things that come to mind is, oh my gosh, you're going to have to undergo chemo. And chemo is systemic chemotherapy, which really remains the mainstay of treatment today, often in combination with newer standard of care agents, but still it's part of the mainstay of treatment. And the issue with systemic chemotherapy, these are extremely potent compounds that are toxic to all cells, cancer cells, and normal tissue as well. And so they are associated with significant off-target toxicities that really limits their clinical benefit to patients, because often these dose-limiting toxicity require discontinuation of the agent. And the classic side effects that you always hear about, include nausea, vomiting, diarrhea, hair loss is just a horrible one for the patient. But the critical ones are immune suppression and heart, lung, kidney toxicities. All of these are significant side consequences of systemic chemotherapy because it's delivered systemically.
What is intratumoral drug therapy, and how might this address some of the challenges of systemic delivery?
Very good question. Intratumoral therapy is essentially what it says. It's delivering drug into or around a tumor. It involves the use of devices and oncologists that have the skill set to deliver drug directly into the tumor. The benefit of doing that, if you have the right compound, is that you're able to deliver a high local dose to the tumor. If you have the right compound, the drug is retained in the tumor, releasing high local concentration, offering the potential for tumor kill and immune priming, tumor debulking and immune priming as well. All of this with significantly reduced toxicity to the patient.
Well, what are the challenges of getting a drug into a tumor and ensuring it stays there long enough to provide benefit?
There are challenges. Tumor accessibility, tumors that are uh, deep in the body as opposed to on the periphery, or the location of the tumor, maybe near a critical structure or under bone, for example. There's um, there's tumor heterogeneity that you deal with any cancer agent. There are pressure gradients when you're injecting into a tumor to make sure that you're able to distribute the drug effectively. And one challenge too, it is tissue pharmacokinetics. I mean, the ability to measure drug across the tumor and how much drug is in the tumor is more difficult with a locally administered drug than it is with a systemic agent where you're just taking bloods in order to measure clearance, the drug clearance over time. I will say, in terms of the biggest challenge, tumor access, um, 10 years ago, the ability to access tumors was really limited to the periphery. We have a discipline that was established in 2016 called interventional oncology that the interventional oncologists, interventional pulmonologists, advanced gastroenterologists, oncology focused urologists now have imaging and procedures that allow access to solid tumors virtually anywhere in the body.
NanOlogy has particle engineering technology that creates stable, excipient free, large surface area microparticles. What's the range of therapeutic classes that you can apply the technology to optimize them for intratumoral delivery?
So we've established the technology in a wide range of small molecules. So taxanes, platins, kinase inhibitors, PARP inhibitors, um, and other classical oncology agents like alkylate agents and DNA transferase inhibitors and those kinds of drugs. Um, the more hydrophobic the small molecule, the better, because the particles will reach a steady state once they're injected into the tumor and then release drug at high local therapeutic concentration for as long as the particles are present. And so the more hydrophobic the particles, the longer the release is over time. We have preclinical and clinical data that demonstrates drug in tumor over 4 weeks, depending on drug class. We have evaluated the technology also in large molecules. We've actually established technology in insulin, for example, as an example of a large molecule. Have evaluated certain immune checkpoint inhibitors like anti-CTLA-4 agents, which are really limited systemically by toxicity.
And what's the significance of these microparticles having large surface areas?
Well, the first is the significance of the microparticle itself, as opposed to being a nanoparticle. So these particles are microparticle size, 2 to 3 microns. That allows for tumor retention. They're large enough so that the particles are retained in the tumor. The increase in surface area is required to allow for high local therapeutic drug release. Um, what's counterintuitive is the ability to make large particles with a large surface area. I mean, if you take a brick and you pulverize it, you significantly increase surface area by decreasing particle size. That's how uh, particle engineering typically works. The uniqueness around our technology is its ability to maintain a large particle size while also significantly increasing surface area. And that underlines a broad intellectual property portfolio that we have around composition of the particles, use, formulation, and technology.
And to what extent are you able to keep delivery localized in the tumor to minimize systemic effects of the drug?
Well, as I've mentioned before, intratumoral delivery historically was limited by access. Access was limited to the periphery. Well, advancements in interventional oncology has allowed now access to solid tumors virtually anywhere in the body. What has lagged are drugs that are optimized for um, intratumoral delivery. Historically, solutions designed for IV administration are what is injected into a tumor, and those we've demonstrated preclinically leak out by simple diffusion. So what's important is tumor retention over a long period of time, and then high continuous local drug release. That's the uniqueness of the particles that we're able to deliver locally, and that's why our drug stays put. We have a lot of supporting data, both preclinically and clinically, that supports that our particles are injected into a tumor where they stay put, releasing drug at high local concentration over a long period of time.
Tumors can be heterogeneous. There can even be variation within a single tumor. How challenging is it to achieve uniform delivery and response?
It's challenging with the administration of any cancer agent, either systemically or intratumorally, because tumor heterogeneity is an issue, and it's a concern that needs to be overcome. The way that we attempt to overcome it is through multiple doses. So we have preclinical data and clinical data. Most of our clinical trials were dose rising and then escalation trials. So we would start with a single dose and then go to more than one dose. And our data tend to be better clinically with more than one dose. Also, preclinically, we've done extensive research that shows generally that more than one injection is better than one injection. And so what we think we're overcoming is with multiple injection is that tumor heterogeneity, because we are able to expose the tumor to drug at some point in the administration process.
And are you able to do anything structurally to control the rate of delivery?
No, we aren't. I mean, what we control is particle size, and that can be controlled and then locked and validated, and then surface area increase. So those two things we can adjust d- during the process of formulation development or particle development. We're changing the geometry of the drug substance. It's the same drug substance, but the geometry is changed so that the particles retain a large particle size with a significantly increased surface area. It has allowed us to date in all of our preclinical and clinical stage assets to take advantage of the 505(b)(2) NDA pathway. Despite having composition of matter patent on the particles, it offers a new chemical entity-like protection. We're still able to uh, avail ourselves of the de-risked 505(b)(2) NDA pathway.
This technology can be applied across a wide range of drugs and solid tumors. What's the thinking in terms of prioritizing indications you'll pursue?
Well, we've completed phase 1, 2 trials in lung, pancreas, bladder, prostate, peritoneal, and ovarian cancers. Um, all of these products are available for partnering as we enter into late phase clinical research. From an internal prioritization standpoint, we looked at unmet need, market size, clinical research intensity, the ability to enroll, the strength of the data that we generated to date, and importantly, whether we can identify an early meaningful surrogate endpoint, because that will allow us to generate objective data more quickly in the ultimate hope of identifying a pharma partner to help advance our assets to the market and patients in need.
And how do you go about selecting which agents would be best to apply your particle engineering technology to?
Well, I wish I could say there was a grand design, but there really wasn't. We established NanOlogy in 2015 with the acquisition of the particle engineering technology, which we call Precision. And with that came a clinical stage asset, LSAM paclitaxel, that had completed a phase 1 trial in peritoneal malignancies. Um, the company that I've been with for a long period of time, called DFB Pharmaceuticals, is the majority shareholder. We acquired the technology. We also own and operate an API development and manufacturing facility that produces commercially paclitaxel API and docetaxel API. So docetaxel made sense to us as a second asset. We've been in discussions on a research collaboration in a pediatric indication, and cisplatin has been used widely in peds, so that made logical sense to us to expand as our third uh, 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 investigational drug. And then we've taken a step back, looking at the platform technology, the precision platform technology itself. As I mentioned before, we established the feasibility of making particles in a wide range of small molecules.
Your large surface area microparticle, or LSAM paclitaxel, is your lead experimental therapy. What's known about its safety and efficacy from studies you've conducted to date?
Well, with LSAM paclitaxel, we've completed 6 uh, clinical trials, all of them phase 1, 2, single arm clinical trials to establish safety, and to generate signs, evidence of preliminary efficacy. And so, in all, with LSAM paclitaxel across the trials, we treated more than 140 patients. There's not been a confirmed drug-related severe adverse event or treatment emergent adverse event in any of our trials to date. The most common adverse event that we see is transient pain, depending on the site of administration. So if we were administering drug um, to the pancreas, there is transient abdominal pain after dosing. To the lung, chest pain, transient. In terms of efficacy in these trials, um, we've been able to demonstrate clinical benefit. So in all of our trials, we've got interesting signs of tumor response, pathologic response. We've also demonstrated clinically now both TME, tissue and peripheral anti-tumor immunomodulation.
We're in an age of immunotherapies. Most people may not know that paclitaxel has immunomodulatory effects. It has effects on both the innate and adaptive immune system. Does the LSAM formulation amplify its immunomodulatory effects?
Yeah, Danny, you are right. Systemically administered paclitaxel has uh, anti-tumor immune modulatory effects, but those are offset by systemic immune suppression and toxicity. What we have been able to demonstrate, and this is both preclinically and clinically, that by delivering extremely high local drug inside the tumor for a prolonged period of time, and this is specifically with taxanes, we're shifting tumor kill from apoptotic kill to immunogenic tumor cell death. And immunogenic cell death is associated with increasing antigens that cause immunomodulatory effects, immune priming effects.
What's the development path forward?
Well, as I had mentioned, we've completed a total across our two clinical stage assets, LSAM paclitaxel and LSAM docetaxel, 7 single arm trials with excellent safety established and uh, signs of tumor response, anti-tumor immunomodulation, and clinical benefit. Uh, all of these trials, as I mentioned, were single arm in nature. And so our path forward is to conduct a well-designed late phase clinical trial in an identified indication. So that will be a randomized clinical trial that will be a phase 2B plus trial. So 2B or 2B adaptive 2B or 2B phase 3 trial. All of that, we have uh, a short list of indications we're evaluating, asset that we're evaluating to do this, but that will be our path forward for next clinical research.
And how wide a range of LSAMs do you have in clinical development, and how broad a set of cancers are you targeting?
The trials that completed, as I'd mentioned, include lung, pancreas, bladder, prostate, peritoneal, and ovarian. We also have a topical product with these microparticles in them, and we completed a trial of topical administration in cutaneous metastases of breast cancer, which is just a devastating condition secondary to metastatic breast cancer. So all of uh, these assets are available for partnering. We are in a research collaboration on a preclinical asset, LSAM cisplatin, for the stereotactic intratumoral delivery into the brain for peds with a rare pediatric brain tumor.
And how has the company been funded to date?
To date, as I had mentioned previously, the company that acquired the technology is DFB Pharmaceuticals, a Fort Worth-based private investment and holding company that has that built a number of pharma-related businesses over the years, divested of major operations in or about 2012, and took some of those proceeds to establish NanOlogy in 2015. So we're a little bit unique in that to date we have largely, well, it pretty much in total funded ourselves through all of this clinical research and preclinical research. We will be seeking outside investment as we move forward to get into late phase research.
And you've made reference to the fact that you're open to partnering on these. Have you had discussions with either investors or potential partners at this point?
Yeah, yes, we've had relationships established, contact with more than 100 different contacts over the last 4 years within pharma. We maintain a database, a pharma target database where we track our progress. We are currently in active confidential discussions with 8 pharma partners, each interested either in a specific indication or potentially the platform itself. So our near-term plan will be continuing to get to a yes or no with these 8 partners. We have calls or meetings set up between now and JP Morgan Healthcare Week, which kicks off each January in San Francisco. And so we will be conducting a number of partnering meetings at that time with established pharma partners and also new contacts. And so we continue to try to advance um, um, into a collaboration likely a clinical collaboration with a pharma partner that may lead to a broader deal over time. In parallel to that, we are planning a capital raise in late 2026 to advance an indication ourselves in the event that we have an identified partner. And so we're going to be using JPM week in January to begin to introduce ourselves to financial investors. And so that's the status of where we're at now from a investor potential partner standpoint.
It's been a tough few years for the sector, although things seem to be picking up now. I'm wondering, what discussions with investors and potential partners has been like, and does one avenue seem to be a place to get better recognition of the potential value of your assets than another?
Great question, and I concur. The financial investments environment for uh, pharmaceutical, or I'm sorry, for um, pharmaceutical investment has been dry over the last several years. Clinical stage companies, preclinical companies, just investment has gone elsewhere. We see that changing. The timing of our potential capital raise reflects the fact that we see improvement, and that improvement, I think, will continue into 2026. But it's a balance between pharma partners and financial investors. I mean, this was all flipped in the 2018, 19, 2020 timeframe, where there was a lot of financial investment into biotech. And when that was occurring, it was very difficult for pharma to try to license or bring something in because of all the financial investment dollars that were out there. That's flipped, but there's much interest from a pharma partnering standpoint. Obstacles that we've faced historically have been intratumoral therapy is off strategy. That is something that we faced early on, 2018, 19, 20, 21 timeframe, but that sentiment is beginning to change. Intratumoral therapy is becoming more and more interesting from a technology standpoint for the reasons that I mentioned before. Access to tumors now is greater, but also because the need for rational combination therapy to treat solid tumors is going to continue to exist for our lifetime, probably forever. Rational combination therapy will be standard of care. The issue with systemic combinations of chemotherapy plus immune oncology agents, newer standards of care, are the stacked toxicities, which limit effectiveness. So you've got targeted therapies that have tried to address this, but they really haven't lived up to their promise because of off-target toxicities, manufacturing complexities, and things. What we are trying to do is to provide the debulking of the primary tumor plus immune priming to increase the response to IO, which our data suggests, and do that without increasing toxicities. So we believe that there is a pathway, and we believe that there's going to be a lot of interest for intratumoral therapy over the next half decade.
Marc Iacobucci, Managing Director of NanOlogy. Marc, thanks so much for your time today.
Danny, thank you too. I really enjoyed it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.